Hi, my name is Paul Campbell, and I'm one of the co-founders of Aster Bio. By training, I'm a molecular biologist and specialize in microbial physiology, or how bacteria make their living. I'm in charge of our R&D and lead the analytical laboratory services. So over the last several years, there's been a shift in monitoring technologies for industrial wastewater. Traditional monitoring technologies really focused on treating the biomass in your system, the mixed liquor, as a black box. It didn't really look at the types of bacteria and the relative amounts of it that make up the mixed liquor. Over time, biotechnologies have improved to the point that we can look at mixed liquor and identify the different bacteria that are present and their levels, and actually make very useful observations on the operations of wastewater treatment systems. These prices have come down dramatically for this type of testing over the last few years, and now we're beginning to see it used in wastewater treatment processes across the world. Today, we're going to focus on two specific types of testing. One, we call a microbial community analysis. This is kind of like a census, in that it goes out and it tells you what types of microbes are present in your mixed liquor and what relative proportions they're present at. This type of a tool is a great survey tool and can really help with troubleshooting different problems. The second tool we'll be talking about is quantitative PCR, which is abbreviated as qPCR. This is a very specific test that looks at key microorganisms within a system, such as the ammonia oxidizing bacteria or key Archaea that help in anaerobic digesters. First, a little bit of background on what we mean when we're talking about molecular testing and in specific molecular biology. So molecular biology is really the study of the molecular basis of life. In this case, what we're really going to focus on today is the hereditary information or the instructions that tell an organism how to reproduce. This stuff is called DNA and RNA. So these are the types of things that you'll find that Ancestry.com and other companies that sequence people's DNA look at to try to identify where different people come from. We use that exact same technology to try to figure out what types of bacteria are in your system. So the typical bacteria or Archaea has in the middle of it a bunch of DNA, shown here as the nucleoid or circular DNA in the center of the cell. This is made up of 4 different bases, which we call purines and pyrimidines, and they tend to form long strands that run in 2 strands coiled together, the DNA helix. The way these different sequences are put together becomes some sort of a molecular barcode so that you can tell the difference between different bacteria. And specifically for microbial community analysis, we focus on a single gene called the 16S ribosomal RNA gene. This particular gene is present in all bacteria and Archaea, and there's versions of it even in people as well. So it's a very good target for identifying the different bacteria. It's composed of a series of very strict conserved regions, shown in blue at top, and very, very variable regions, numbered V1 through V9. It is the differences in these variable regions that allow us to tell the difference between bacteria 1 and bacteria 2. So when we look at this type of a technology, we'll often sequence maybe 10,000 different genes or 16S ribosomal genes from a wastewater treatment system. That's good enough, but usually we shoot for 30 to 50,000 sequencing reads. The backbone of this technology is a platform that's called PCR, or polymerase chain reaction. It's used both in sequencing and in the quantitative PCR tests. It uses a couple of terms that I'd like to explain very quickly. The first is DNA polymerase. This is an enzyme that basically copies DNA, sort of like a Xerox machine. The second is template. A template is the genetic information that we pull out of the bacteria originally. It's their DNA in a purified form. Primers are very short pieces of DNA that identify the 16S gene and allow us to build more copies of it. And the nucleotides are simply the building blocks that we use to assemble new genes. So how does this work? Shown here is a schematic of what a polymerase chain reaction looks like. Starting from the left, you have in a small tube, you have a whole bunch of components. You have the DNA polymerase that we mentioned earlier, combined with the template, the primers, and the nucleotides. In the first step, you separate out the DNA helix into its two independent strands simply by heating up the reaction to 94 to 96 degrees Celsius, so almost to a boiling point. You then cool the DNA or cool the reaction mix slightly, and this allows the DNA to start sticking back together. But because of its size, the primers stick to the single-stranded DNA first in step 2. In step 3, the temperature is at just the right point for the polymerase to work well. And those primers do what you might expect. They prime DNA synthesis. And you get extension of the primer to copy the DNA using the nucleotides as the building blocks. And then you simply repeat this process over and over. Each time, you double the amount of DNA present in the sample. So how can you use this type of testing in your workflow right now? Well, testing on a regular schedule can be very helpful because it always helps to know what a good operational baseline looks like. We often suggest that people use microbial community analysis on a quarterly basis or whenever they're going to make a large operational change. Then, if there's key specific microorganisms that you want to track for your system, we recommend using qPCR or quantitative PCR on a more frequent basis. Most often, people will use outside labs for these services. First, because it requires some training for them, and understanding how to troubleshoot those reactions are a bit challenging. And labs can do this sort of testing. So running the tests is not that challenging, but the difficulty is really interpreting the information and making it useful for your specific system. While this wastewater molecular diagnostics can provide insight into different parts of your plant, such as biological nutrient removal or foaming and bulking events, it's also useful for optimizing chemical usage and energy usage. And shown on the right here is just an interpretation of the data of a particular municipal wastewater treatment system. As it stands, this type of information is a little bit complex, but I'll explain more as we go along. The system is really quite simple. The operator goes out and collects a small sample of mixed liquor or other biomass from the system, puts it into a container and ships it into the lab. The lab then extracts the DNA, performs QA/QC analyses, and then sequences the DNA. This generates something along the lines of 20,000 or more data points that we call sequencing reads. The bioinformatics software that we use then compares these sequencing reads to a public database to create an output of each read that identifies it by what type of bacteria it is. Then comes the data interpretation. Generally, for wastewater systems, it helps to have someone who's focused on wastewater to interpret the data. And this really helps if the person's seen multiple plants so that they can compare your data against other plants and identify best interpretations. So what does the data really look like? We mentioned 20,000 sequencing reads or more. This is the raw output, an individual sequencing read. And honestly, even this doesn't mean much to an expert. It requires further processing by the software. That processing, though, allows us to know what types of bacteria are present and in what relationship. And that's where we see here in what the chart on the right that I showed earlier that we call a Sankey chart. It covers the top, in this case, the top 30 bacteria present in this particular municipal wastewater system. And it can help interpret where different problems are. For example, there are on this slide, or pardon me, on this chart, there are a couple of bacteria to point out that show some interesting aspect about the plant. First, you'll find that up near the top, you see Nitrous amonis, and near the bottom, you see Nitrospira. Those are the ammonia and nitrite oxidizing bacteria common in many systems. The other thing you'll see near the middle is Trichococcus and Thiothrix, the presence of filaments. So this tool can help identify nutrient removal issues and identify filaments in the system very quickly. But interpreting this data can be a bit of a challenge, so we've simplified the system and grouped bacteria together so that an operator can interpret it very quickly. Here is an example of the output. Zooglea, for zooglial bulking, is a common bacteria you'll find quite often. And it's included in the, well, I'm going to hope you edit this out and I'll back up. So we try to group our bacteria by function so that you can interpret the results very quickly. Shown across the top here are the different categories of bacterial function that we often find in a system. And on the left side are the bacteria. So from this chart, you can tell that you have Zooglea present, and they happen to be nitrate reducers as well. We, from experience, know that this level of Zooglea, 6.5%, is moderate and probably not associated with bulking at this time. You also see the amounts of Nitrospira and Nitrous amonis that are present. These are very common levels for nitrate and ammonia removal in most municipal systems. qPCR, the other test we discussed, is a rapid test. It takes only an hour and can often give data back the next day. It's used to detect very specific organisms of interest, such as the ammonia oxidizing or nitrite oxidizing bacteria, specific filaments known to be in your system, nonfilamentous bulking, and even sulfur-reducing bacteria. So how do you take lab testing and bring it into daily operations? Well, the first step is to get the data back in a reasonable time frame. If you're trying to make an operational decision, you can't wait weeks for data. Therefore, you need a lab that can provide the data in a short period of time. Routine microbial community analysis can be turned around in 7 to 10 business days. And if there is an emergency, it can be rushed within 48 hours and sometimes even within 24 hours. Quantitative PCR, on the other hand, takes only 2 to 3 days and when rushed, can be returned within 24 hours. It's important for the reporting to be designed for people who are not molecular biologists. For example, when you go to the doctor and he does a blood test, he doesn't simply read off the numbers to you. He helps you interpret how those numbers impact your health. Likewise, you should expect the same thing from the laboratory that you use. They should be able to tell you how the data can be used to impact your operations. Example applications include monitoring changes in the system when you're changing operational conditions. Troubleshooting challenges such as loss of nitrification, and also trying to identify chronic toxicity events, which we'll discuss further in a minute. So monitoring ammonia oxidizing and nitrite oxidizing populations. Why are we interested in this? Well, the main reason we're interested is these are slow-growing bacteria that are also very sensitive to upset conditions. Even just small changes in the chemicals in the influent can kill them very quickly. Usually, the first sign that the plant is having a problem is that at the end of the plant, at the effluent, you see ammonia or nitrite coming out. Using quantitative PCR, you can track and watch the changes in these bacterial populations and see them start to decline before you see the impacts on the effluent. As an example, here's a case study from three different plants. On the left is a healthy chemical plant where nitrification is at typical levels. In this example, we're showing nitrous amonis as the ammonia oxidizing bacteria, and nitrospira as the nitrite oxidizing bacteria. Hyphomicrobium is included because it's commonly found in most systems, and it's present at a pretty consistent level. And if a system has lost nitrification, as in the two examples on the right, the real question is, why have they lost nitrification? In the middle case, nitrification disappeared, but the bacteria were still there at low levels. This was an example from a Houston facility where Hurricane Harvey flooded out the system. Using quantitative PCR, we were able to identify the fact that the bacteria were present, not dead, not absent. They needed more dissolved oxygen to start growing again. We suggested to the operators that they supplement with hydrogen peroxide to raise the residual dissolved oxygen, just a few ppm, and nitrification was restored in just a few days. In the example on the right, with chronic toxicity, they lost nitrification for weeks on end with no real apparent cause to it. Every time they would add additional nitrifiers, they would restore nitrification for a day or two, but then checking again, the nitrifiers would be dead. Further research identified metals toxicity in the system that needed to be removed. Once the metals toxicity was eliminated, removed from the influent, nitrification was restored. Nonfilamentous bulking is another major problem in many different systems. It's caused by extracellular polysaccharides produced by different bacteria, especially Zooglea and some close cousins that are typically found in most wastewater treatment plants. India ink on a slide stain is the typical way people identify this type of a problem, but it can be challenging to understand and interpret what's causing the bulking event just from the slide. Here's an example of two facilities where one was experiencing bulking and the other was not. Zooglea and Thyera are the major causes of non-filamentous bulking in most plants. They're both really key because the extracellular polysaccharides do help the flux stick together, but if it's present in too large a quantity, it causes the bulking issues. Wastewater treatment plant 1 here was experiencing bulking with very, very little space in the clarifier. The wastewater treatment plant number 2 was operating normally. And unfortunately, the India ink stains were being inconsistent and making it difficult to tell the differences in the Zooglea and Thyera levels in the two different plants. However, with quantitative PCR, you can tell that there was very serious bulking due to Thyera in plant number 1, while plant number 2 had much lower levels. Following the trend in these bacteria over time, you can predict when you will begin to see bulking issues. Filaments are another big area where quantitative PCR and sequencing can help. Often, you look in a microscope and you see a whole bunch of filaments, but they're not really always readily identifiable. our molecular tools, we can identify and track changes in these filaments very quickly. Here's a recent example of the filament Thiothrix and how it changed over just a 1 month period of time within a plant. It went from about 3.6% of sequencing reads, as shown in the top, to about 13.5% of sequencing reads, as shown at the bottom in April. And in this period of time, the plant noticed that the increase in Thiothrix caused real operational issues. Had they been tracking on a more frequent basis, perhaps weekly, they might have been able to catch the Thiothrix levels increasing before it caused major problems. As a final example, anaerobic digester testing. We do test Archaea as well, although we've spent most of the time talking about bacteria. Recently, there was an anaerobic digester that was not performing well. It was producing very low levels of methane, and they didn't understand why. When sequencing, we were able to identify that the major Archaea present in the system is called Methanothrix. Methanothrix is a filamentous Archaea that uses a different pathway for producing methane than the other bacteria. It's this presence of high levels of Methanothrix that caused the anaerobic digester to produce very low levels of methane. Thank you very much, and I appreciate your attention.